Thank you very much. And so the topic is lamb diagnostics for detection of coconut litter yellowing type diseases in Africa. And I'm presenting on behalf of Professor Matthew Dickinson. So this will be the outline of the presentation. I'll give a background. I'll talk about the comparative analysis between PCR and real-time lamb. I will talk about the development of assays that discriminate LY strains in Africa. And then I'll move on to optimization of a rapid DNA extraction protocol from coconut trunk boundaries. And then a development of an internal control assay for insect samples. So by way of introduction, the litter yellowing type disease is very prominent in Africa. I think as Wayne alluded to, this is a graph, this is a map adapted from Egya et al. And we can see that the disease is present in Cote d'Ivoire, in Ghana, in Togo, in Nigeria, and then in Cameroon. And as Assunta mentioned, we now have it also in Equatorial Guinea. And when it comes to the eastern part of the continent, we have it in Mozambique, and we have it in Tanzania, and we have it in Kenya. So the disease is very prominent in Africa, and it has destroyed millions of farms and destroyed the livelihoods of many resource-poor farmers on the continent. Different strains of phytoplasmas are involved with the disease in different parts of the continent. And the use of fast, reliable, and cheap diagnostics are an essential part of the disease management strategy. So for conventional phytoplasma diagnostics, there are no culture-based techniques or simple microscopy methods for detecting the presence of phytoplasmas in plants or insects. The conventional method is the PCR technique. However, the thermocycler is bulky and is not amenable to in-field use. In parts of Africa, the farms are in very remote locations. Sometimes you don't have electricity in those parts, so you want a technique that is amenable for in-field use. So real-time lamb, coupled with the rapid DNA extraction protocol, is considered as a potential method that can be used for phytoplasma detection. And very simple equipment have been developed which can be used in remote parts of the continent. So in this equipment, usually the data can be stored and then transferred back when you are in the lab. So the objective of this part of the study was to develop fast, reliable, and validated laboratory diagnostics for the litter yellowing type phytoplasmas in Africa. And then the second part was to apply these techniques in the field. So what we have done, firstly, is to compare PCR and the real-time lamb. And there is a published lamb primers for the litter yellowing disease in Ghana. This was published by Tomlinson et al. In 2010. So for the samples we have collected in our nationwide survey, as I mentioned in the other presentation, we have compared PCR and lamb, the ability to detect the phytoplasma. And generally we saw that with the exception of the early stage infection, that is stage 1, and in the symptomless, at the symptomless stage, there was generally consistency between nested PCR and lamb. It's only at the first stage and the symptomless stage that nested PCR was more sensitive in detecting the phytoplasma than in lamb. Lamb, however, was more sensitive than the first round PCR in most instances. All right. So we now tested this lamb assay against a broad range of phytoplasmas, and this included the phytoplasma associated with the disease in Mozambique, which belongs to the 22 group A. And we also tested them against samples from Mexico, from groups 4A and D. And from the results, we realized that the assay, which was specific for the Ghanaian strain will amplify the Mozambican phytoplasma, the Mozambican disease with equal efficiency. The assay, however, did not amplify samples from Mexico. And this is typically the output you get from the real-time lamp. Now, having validated the assay in the lab, we went out into the field to test the method in the field. And as I mentioned earlier, for lamp to be used in the field, you need a rapid DNA extraction method. And there's a method that has been previously developed. It takes about 2 minutes. And there is this commercial buffer which comes in a small bottle with steel ball bearings. So what you do is you put about 0.5 grams of the plant material in the bottle and shake it for about 2 minutes. And then you can take about 2 microliters and put it directly in your lamp reaction. The remainder of the contents in the bottle, the plant material, can be stored and later extracted in the lab or stored for long term. So we use this lamp machine, the lightweight portable lamp machine, which is battery operated, out in the field. And what we do in the field is to want to run concurrently an internal control, which is specific for the plant DNA, that's the coconut DNA. And so we want to make sure that the extraction protocol is efficacious in the first place and that our results are not false negative results. So typically we run the 2 tests alongside, first with the one giving us an output for the plant primary, what the coconut DNA is there, and then the phytoplasma DNA. So we have done this in remote locations in Ghana. And what we do is to already prepare our mixes in the lab, take a cooler with ice pack, and then take the reaction mix, everything is already set. And in the field, all that we do is to use our rapid DNA extraction protocol and then run the test in the field. So we can run the test on your carbonate or on the floor, maybe on the cardboard. All right. So we have then moved on to develop assays that discriminates between the group 22 phytoplasma, the LY strains, based on a leucyl tRNA gene. Now, generally, we know that the LUS and the 50S gene is known to give more sequence variability between phytoplasma groups and subgroups than the 16X to 23S ribosomal RNA gene. So we developed new primers based on the 2 genes, but we found the LUS assay to be more effective. And the LUS assay was able to discriminate between group 22A and group 22B, that is samples from Ghana and Mozambique. And the assay did not amplify samples from group 4A and D, samples from Mexico, or samples from any other 16SR group. So those are typically the validation output. This and the first primer, the CSP, is a published primer which detects. We realized that it would detect the group 22A and the group 22B, and it does so in about 17 minutes. All right. And these are the 2 sets of primers we have developed in this work, and these are based on the LUS gene. Now, this assay was able to amplified samples from Ghana, but not samples from Mozambique. And one of them was very, very efficient and could amplify the samples within 10 minutes of running your reaction. And these assays did not detect phytoplasma from any other 16SR group. So we decided we went on to optimize the rapid DNA extraction protocol. Now, the original protocol, which was developed by Tom Linson et al., is a bit cumbersome in the field. After getting your coconut trunk borings and shaking putting it in your commercial buffer, the original assay, you had to get your LFD strip pipette about 80 microliter of the extract onto it, wait for a while for it to dry, and then cut a strip and put it in your lamp reaction. It was a bit cumbersome to do in the field, so we decided to optimize the protocol. So what we have done is to have tested a number of buffers, including T buffer, CTAP, PEG, for their effectiveness as an in-field extraction buffer. And a PEG buffer that has been published for extracting rapid DNA extraction protocol for other tissues was found to be very effective. What you do is to take a little bit of sample, about 20 milligram of coconut stem borate, put it in your tube, and then you can use the micropistol to just mash it in the tube. And within 30 seconds, your DNA, your extract is ready to be used in your real-time lamp assay reaction. And then this assay has been validated using coconut stem borings, insects, and then leaves from other plant tissue. So my colleague, Matt, has been trying this in different places around the world. So after improving the rapid DNA extraction protocol, the team was supposed to have come to Ghana for us to work in the field, but due to COVID, they were unable to do so. So field simulations were done at the University of Nottingham, and using the rapid DNA extraction protocol and the real-time lab, and with the samples that were collected during the nationwide survey. And generally, the assay was very effective, and it amplified the samples. The maximum time we had an application was for 34 minutes, and assay was run together with the Cox assay to detect plant and the plant DNA, and to ensure that the extraction protocol was efficient. Now, we also aimed to develop an internal control assay for insect, just like we use Cox as an internal control for plant DNA. So there is a published primer for invertebrate insects. So what we did was to try it on insects that we had collected from Ghana. Matt Dickinson did this from the UK. And what we observed, what we found out was that generally the assay was ineffective in amplifying the insect DNA. So we had to tweak the assay a little bit. And what we have done was to take the F3 and then B3, the outer primers, and then use it on the insect DNA, amplify them in PCR, and then to now redo the internal primers, that's the famous and the backward loop primers. And these assays are now being fine-tuned for use as an internal control for insect DNA. In the meantime, the protocol we use in the field is to spike a control. We spike a control with insect DNA, and this works out very fine, whilst we fine-tune the assay as to be used as an internal control for insect DNA. So in summary, we have developed RT protocols for in-field diagnosis of litter yellowing, and this assay has been validated. We have an assay that is able to discriminate between the group 22, the two subgroups, that is subgroup A and then subgroup B. And the rapid DNA extraction protocol has been fine-tuned. It can be done within under a minute, and the internal control assay for insect DNA is being fine-tuned. Thank you very much for your attention.